Post-transcription is the very special process that only happens in eukaryotes.
Why post-transcriptional event is so important for eukaryotes? Because this process to modify primary transcript prior to protein translation. The post-transcriptional events are capping, RNA slicing, polyadenylation, coupling of transcription and RNA processing with nucleus, RNA transport, and finally, translation.
The first transcriptional event is capping.
There are three types of enzyme involved in capping. First, RNA triphosphate removes leading phosphate group from mRNA 5-terminal triphosphate group. Second, guanylyl transferase known as capping enzyme added GMP to produce guanosine cap in a process called guanylylation. Third, guanine 7-methyltransferase methylate the guanosine cap and attached to 5 of transcript.
Why we need capping?
Capping that protects mRNA from 5-exonuclease activity and it prepares the transcript to interact with IF2 to position the mRNA on ribosome.
How these enzymes works?
First, Y-phosphate at 5 end of pre-mRNA is removed by phosphorhydrolase. Then, 5-5-linked guanylate residue added to 5 end by guanylyl transferase. Then, the association of capping enzyme with RNA polymerase 2 through phosphorylated CTD methyl group is then added to N7 of 5-guanylate and 2 hydroxy groups of the nucleotides in primary transcript, the 5 end with cap now.
Second is RNA splicing.
In RNA splicing, the introns are excised and the exons are sewn together to form mature mRNA, small nuclear ribonucleoprotein particles called as snRNPs, which is also pronounced as SNRPs, that are involved in splicing. A snRNP consists of a small RNA which are 100-200 bases long and about 10 different proteins. The snRNP form lariat, which is a covalently closed loop of RNA, is formed by attachment of the 5' of the introns invariant 5G to the 2O at the branch. This branch site is essential to splicing. Then the exons join, excising the lariat.
Third is polyadenylation.
CPSF, CSTF, CF1, CF2, PAP, and PABP2 are the series of enzymes and factors that involved in this mechanism. CPSF binds upstream of AA at 5 end. Then, CSTF interact with downstream Q sequence and bound with CPSF to form loop in RNA. PAP stimulates cleavage at polyacide, binds to AD12 as at 3O group at slow rate. PABP2 accelerates addition of a bipap and signal PAP to stop polymerization after addition of 200 to 250 residues.
Fourth is coupling of transcription and RNA processing.
RNA polymerase 2 supports coupling of transcription and pre-mRNA processing by the work of CTD called as carboxyl terminal domain, acting as landing pad to enhance mRNA maturation. CTD stimulates capping, splicing, and polyadenylation processes, which has been proven in vitro.
Fifth is RNA transport.
Each RNA have different transport pathways through nuclear pore complex as mentioned in the table. This process mostly involved exporting pathways, which is signal mediated by nuclear export signal called as NES. NES RNA binding protein needed to export all RNA except tRNA. All RNAs except mRNA are dependent on RA and GTP, small RAS-related GTPase. GTPase enzyme binds to GTP to hydrolyze GDP and release energy.
Sixth is translation.
In translation involves initiation, elongation, and termination. In this process, mRNA translated to protein, which is known as end product via ribosomes and tRNA. From the mRNA sequence, each 3 nucleotides codes for 1 amino acid. The final product of translation referred to as polypeptide.
Thank you.